Kia ora koutou, everyone. My name is Jane Barnett, and I'm delighted to be able to introduce today's Weber Training Teleclass lecture, which is the final lecture for the 2025 Australasian series. Today's speaker is talking to us from Western Australia, and he's contributed to Weber Training in the past, so it's great that he was able to take up our invitation to return once again. Professor Tom Riley is based in Perth in Western Australia, and he divides his time between PathWeb Laboratory Medicine as a senior clinical scientist and the University of Western Australia as a professor in the School of Biomedical Sciences. He's had a long-standing interest in healthcare-associated infections, and particularly in relation to the diagnosis and epidemiology of Clostridium difficile infection. He's a fellow at the Royal College of Pathologists, the Australian Society for Microbiology, the American Academy of Microbiology, the Society for Healthcare Epidemiology of America, and the Faculty of Science at the Royal College of Pathologists of Australasia. So with that very impressive bio, it gives me great pleasure to introduce Tom, and who is really recognised as one of the experts in relation to C. diff infection. Today, he's going to be speaking on the topic of Clostridium difficile, One Health, and the rise of community-associated infection. Thanks very much, Tom. I'll hand over to you.
Thanks, Jane. That was actually last year's title, but if you have a look at the screen, you've got this year's title, which is what can knowing something about the evolution of C. difficile teach you about infection prevention and control? And for those of you that think I should be calling it Clostridioides difficile, both of these names are still validly published, and you can use whichever name you prefer. And I've always preferred Clostridium difficile. So anyway, thanks Jane and Paul for the invitation to come back again and talk about my favourite organism. So this is, I always feel obliged to put up some sort of outline, but I'm not very good at following my own outlines, but this is some of the things I'm going to talk about today. If you listened to my talk last year, I think almost exactly a year ago, in fact, some of the slides will be familiar, but I'm kind of going to embellish them a little bit. And in fact, in some ways, go back in time, literally go back in time, and in other ways, look forward to what we might expect and what can we learn from the evolution of this organism. So I like starting with a definition slide, and I did show this one last year. I think we can forget the top part of it, which is the distinction between community associated and community acquired. And you'll see that there's Cliff McDonald's surveillance recommendations from 2007, so they're kind of out of date. So let's not worry about those. I'm sure you all know about them. I just want to emphasize the point or the difference between the source of infection and the reservoir of infection. And so the reservoir is where the organism actually lives, it resides, metabolizes, multiplies, whereas the source is just a place where it might come from. And of course, a reservoir may or may not be a source, but you do need to make that distinction, particularly when you're talking about C. difficile, because people talk about sources of C. difficile as the reservoir, but really the only true reservoir of C. difficile is the gastrointestinal tract of now a huge range of different animals, and I'm including birds and reptiles and things like that. So let's go back in history. I'm sure I've showed this slide a million times. So this is sort of early 1970s. This is what we thought about C. difficile. So there's a hospital. The hospital's got C. difficile in it. Patients in the hospital take antimicrobials. And you need to remember that at any time, 50% of the patients in a hospital will be on antimicrobials. So some of those patients developed diarrhea. Back in the 70s, that often progressed to pseudomembranous colitis, which you can see on the bottom right-hand corner. And then back then, the only way to deal with this disastrous situation where you've got pseudomembranous colitis, which is kind of at one far end of the spectrum of disease that C. difficile can cause, was to remove the affected part of the gastrointestinal tract, which is the large bowel. And you can see one of those there. So the important question that nobody seemed to want to ask back then, and indeed they don't ask now, is where is that C. difficile? Where did it come from in the first place? It hasn't been living in hospitals for years and years and years. So really what I want to talk about, the underlying theme, or one of the underlying themes for today, is where does the C. difficile come from and how does it get transmitted to humans to cause disease? So we know things are important when eventually the CDC puts out one of their reports, and this was I prefer using their 2013 report, where it appears, at least on this list, that C. difficile came out as their #1 urgent threat organism. And at that stage in the United States, they were seeing 250,000 infections a year, 14,000 deaths, and perhaps even more importantly, it was costing a lot of money. And they did make the point that C. difficile infection was occurring in hospitalised or recently hospitalised patients. So the numbers in the US have changed a little bit. They've come down a bit, as you'll see in a second. So now we've moved to the 2019 report, where we've got a couple of organisms added to that list. C. difficile is still there with its new name, Candida auris. I'm sure Candida auris has got a new name as well, but I can't remember what it was or is. So let's move on to some of the contents of this 2019 report. The numbers had dropped slightly, and that's because, as they say on this slide, healthcare-associated C. difficile infections were decreasing, but community-associated infections were not decreasing. In fact, they were increasing. And there's a couple of things that they've said, which they've said, for example, that this organism, Clostridioides difficile, was previously Clostridium difficile. It is still Clostridium difficile, and I have that fight with journal editors every week. So it's still a big problem. It's still costing a lot of money, and indeed, it's the most common healthcare-associated infection in the world. So a little bit about C. difficile. It's a very ancient genus. It's hundreds of millions of years old, and it's an anaerobe. And those of you that have looked at the history of the world will know that for a long time ago, the world was completely anaerobic. And so it kind of makes sense that some of the very early organisms to emerge were indeed anaerobes, and C. difficile is one of those organisms. The population structure of C. difficile consists of at least 8 clades, and I'm sure there are going to be more clades discovered, and I'll show you some phylogeny in a minute. The 2 major genes for toxins, which are the major virulence factor, are located on this so-called pathogenicity locus. There are non-toxigenic strains that don't have a pathogenicity locus, and they don't produce toxins, and they don't cause disease. The the paylock, as we call it, is always found in the same position on the chromosome. And so initially people decided that the pathogenicity locus was integrated into the chromosome before we had this diversion and a number of different clades started to appear. But the evidence says otherwise. And so we have non-toxigenic strains, bit of confusion between ribotype and sequence type. And I won't go into that, but it complicates the issue, as we'll see in a minute. But the really interesting thing is that this irregular distribution resembles a mobile genetic element. And there's some quite good evidence that the toxins have got into C. difficile via a bacteriophage, because there are bacteriophage genes you can find in some strains of C. difficile. Some have disappeared completely, but in some strains they're still around. So, press that one. So here's the world 300 million years ago, so the supercontinent Pangaea, and you can imagine that whatever crawled out of the very large ocean at the time onto this continent was roughly one strain of C. difficile. And then as the continents moved, drifted, this is what happened over a number of millions or hundreds of millions of years. Pangaea broke up initially into Laurasia and Gondwana, and then down on the bottom left corner there you can see something starting to emerge that looks a bit like the world today. And you can also see there that India floated around for many millions of years before it banged into the rest of Asia and created the Himalayas. So my theory is that in India there would have been a separate group of C. difficile. So using whole genome sequencing, we can categorize C. difficile as shown on this slide. And the reason that there are much more or many more of those pale blue strains is because that's clade 1. And so most of all this early whole genome sequencing was done in the United Kingdom, which, as you'll see on one of my next slides, is probably the home of clade 1. So it's kind of understandable that they would have more clade 1 strains there. And that's one of the big problems with whole genome projects like this, is that the results that come out of it are only as good as the bugs that you put into it. And that has created some problems. So what you can see there, if there's a black line coming along, that means that it's non-toxigenic. But when it's got a coloured line, it means it's toxigenic. So you can see in clade 1, for example, there are toxigenic strains of C. difficile and non-toxigenic strains in C. difficile. If you look at clade 2, you see a much smaller group of strains, but they're all toxigenic strains. You look at clade 3, all toxigenic strains. You look at clade 4, there are more non-toxigenic strains than toxigenic strains, and the same applies for clade 5. And then when we did this study with people in Oxford, we took over some Australian strains, and lo and behold, many of those didn't fit into those first 5 clades. And that group at the very bottom of this picture is a group that we call cryptic clades. And that was the first cryptic clades. And they're all Australian strains that had never been looked at before. So you can see here some interesting facts. Just the two top pictures. The one on the left, one says A, so that's clade 4, and you can see the scale on the bottom is years. So you can see that clade 4 actually only acquired a pathogenicity locus about 500 years ago, and equally in the one that's got a B on it, that's clade 1, and that's just part of clade 1, which shows that there was pathogenicity locus loss about 50 years ago, and then you get this whole subgroup that are non-toxigenic. And this is all done with a very clever program that I'm not going to talk about, other than say it's called backdating, as the name kind of suggests. You can work out how old strains of C. difficile are. And that was developed by a very smart French bioinformatician, whose name is at the end of the reference there, and that's Xavier Didelot. And he's a very smart guy. So another smart guy is Dan Knight, who's a PhD student of mine. And so we looked at a group of strains. In fact, we actually downloaded all the available genomes that were in public databases around the world. And then this exercise here is to look at whether they all, to look at the differences between the clades in terms of their average nucleotide identity. And so the rule is that if you fall within 96% the same average nucleotide identity, you're in the same species. So if you look at along the top there with the red dots, you can see that, the blue dots, I'm not colorblind. The blue dots, clade 1, probably the biggest representation, as I said, for the reason that most of the sequencing has been done in Europe. Clade 2, clade 3, and clade 4 all fall well within that 96% line, but the clade that sits right on that line is clade 5. And so the interesting thing here is, do we know whether clade 5 is moving towards the other clades, or is it moving away? And so below that line, you get the now 3 cryptic clades. So you can see on that 91% line, most of those are those C1 cryptic clade that we found earlier in that study that we did at Oxford. So they're clearly not C. difficile as we know C. difficile to be. And at some stage, they will be given new names. But I should say that, A, they have very odd toxin structures, but B, they are being isolated from patients who have infection. So some of them do produce toxins. And eventually you'll have to learn some new names. So this is just representing that first phylogenetic tree that I showed you in a slightly different way, showing that the cryptic clades are some distance. And in evolutionary terms, for example, you've got the first 4 clades that you can see there, and clade 5 is some distance. Now, in evolutionary terms, that's about 80 million years away. And interestingly, as you'll see on one of these slides coming up, I think that clade 5 actually lives in Australia, and clade 5 separated in Australia, separated from Gondwana about 80 million years ago. So that kind of fits in. So this is a little bit more complicated. It's from that same paper of Dan Knight, published a couple of years ago. And I'm not going to go through all this because it looks very complicated, and I can assure you it is. But the point that we're trying to make here, again, this is using Xavier Didelot's backdating program. what that tells us is that the cryptic clades actually emerged millions of years before the clades that we are more familiar with. So they've been around a long time, and we need to do a lot more work on those to understand the evolution of C. difficile. So this is, I coined this term phylogeographical tropism to hopefully imply that certain clades lived in certain parts of the world. I think that can be very useful. The only problem is we've done so little work on C. difficile in Africa. We don't really know what's going on there. But my suspicion is that is in fact the home of clade 3. And we don't really know what's going on in South America for exactly the same reason. And the other problem is that this large blue area there, which I've called Asia, it's huge. And really we only know a little bit about what's going on in Southeast Asia, but all the evidence says that Southeast Asia is the home of clade 4. And the circled area, of course, is India, where I suspect we will find eventually a different clade. Now, you have to remember, though, of course, that all this is becoming very murky with travel. People travel, and as I'll make the point in a minute, travel of animals. The only way that organisms get around the world is with population movement. It's not the only way, of course, because a single organism can travel with a single person. But if you want to get major changes come with population movement. And we always think about human population, but we move lots of animals around the world, as you'll see in a minute. So just to give you a quick idea of how different things are. So this is a project we did with some a Japanese pharmaceutical company a couple of years ago now, showing you what I'm trying to show you here is the difference between Australia, which is down there, and you'll see that in Australia we predominantly have a strain called ribotype 014 or 020, which is a clade 1 strain, and has probably come from Europe, which just reflects our history. We were very much English for many years, but we've also found in Australia many new strains that we cannot identify, which are clade 5 strains. And those other circles are Asian, and the big difference there is you'll see most of those have lots of ribotype 017, which is a strain which we think has evolved in Asia. And you can see those Asian countries there with large red, bright red, as distinct from ribotype 070, which we have a bit of in Australia, which is a different red, not a good choice of colours. So you can show the difference in population between these areas of the world. And so whole genome sequencing and bioinformatics could allow us to track the emergence of ribotype 027, which was a very nasty strain which emerged in the United States and Canada in the early 2000s. And you can see that there were 2 strains that emerged that were fluoroquinolone resistant, and one strain didn't travel very far. I've always called it the American strain, and my feeling is that Americans didn't travel very far around about that time, whereas the Canadian strain, as I've called it, travelled a lot and certainly travelled into Europe and caused massive outbreaks in the UK and then subsequently in continental Europe. And it did get transported to Australia. There was a cluster of cases in a hospital in Melbourne, but ribotype 027 did not and has not established itself in Australia. We get the odd sporadic case, but what that tells you is that Australia doesn't have the right conditions. Ribotype 027 is a North American strain, and there's something about North America that ribotype 027 likes, and so it didn't like Australia. I can't imagine why anybody wouldn't like Australia, but anyway, ribotype 027 didn't like Australia, and the end of that arrow there is not where Melbourne is, of course. The geography instructors in this particular paper didn't do a very good job. So whole genome sequencing can tell you a lot about what C. difficile is doing. And the way we do that is by looking at the acquisition of single nucleotide polymorphisms. And C. difficile mutates quite slowly, roughly somewhere between 1 and 2 SNPs per year. So you can look at a collection of strains and determine if they're related by looking at the number of the differences in the number of SNPs. And it's a very accurate way of determining relatedness. And so my feeling is I'm a big fan of whole genome sequencing, but really for 2 reasons. One, it can give you a sequence type, which is better than a ribotype, which is kind of the main typing method these days around the world. And it can tell you whether 2 strains are related and how related they are. And in terms of outbreak investigations, that of course is very important. So a couple of old slides. These are slides I got from the anaerobe reference laboratory in the UK many moons ago. But it just, this is when ribotyping first started. So ribotyping was actually developed by an ex-PhD student of mine when she went to the anaerobe reference laboratory to work. And so she started this PCR ribotyping method of typing C. difficile and just gave them numbers. And at the time when she started this, there was a bit of an outbreak in the UK of C. difficile. And so clearly that strain, which was named ribotype 1, because it was more likely that that would be the first one that she got because it was so prevalent, and the numbers just continued. And so we're up to about 800 different ribotypes. So the 2 other interesting features of this, and that is the second most common strain there is type 106, which is a strain which has gone, really gone around the world, moved to the United States, where it is, I think, still the most prevalent ribotype of C. difficile. And it got to Australia about 10 years ago, and its numbers are increasing. And the other really interesting strain on this particular slide is right at the end there, and that's ribotype 023. So that's a nasty binary toxin-producing strain, causes severe disease, and my suspicion is and it's in clade 3. So my suspicion is that it's, and there's good evidence of it now with what we're seeing in some European countries where ribotype 023 is causing much more disease. I think there's quite good evidence that the migration from Africa into Europe is bringing this ribotype 023 strain into Europe. And then a couple of years later, when ribotype 027 has got to Europe, you can see here in the UK, type 106 has gone up, as it has been around the world. 027 gone up as well, prevalence around 25–26%, and decline in type 001. And this is what happens really with all organisms. Certain organisms come along and replace whatever's floating around. And clear example here, 027 came into Europe, caused massive problems, and then has disappeared. because 027 is not a European strain. It's a North American strain, and it's still floating around in North America. And so if you know what sort of strain you're dealing with, you'll know what its toxin profile is. And so that's why it's worthwhile knowing what these strains are. And you can see here that, for example, clade 2 strains, and in this bottom panel, you can see that's B, toxin profile, A positive, B positive, binary toxin positive. And C. difficile strains that produce binary toxin as well as the other two toxins are invariably invariably cause more severe disease. And so one needs to be on the lookout for those. So knowing something about the strains is important. And at the moment, the best way of doing it is whole genome sequencing. Unfortunately, this is expensive, and it requires proper trained bioinformatician to analyze the data, because whole genome sequencing produces huge amounts of data. And what we need is somebody to fund all this whole genome sequencing so we can understand all this better. So, and then the other problem, as I said, if you don't really understand what's going on, you end up making mistakes. And my name is on this paper, even though I urged Brendan Wren to take it off, because I wasn't happy with his conclusion. And this is a study where he concluded, or I guess they all concluded, perhaps, that they thought that ribotype 017 had emerged in North America. Well, that's complete rubbish. Ribotype 017 is an Asian strain, and there's plenty of evidence of that. And we'll see some of it on these next couple of slides. So this is some work done by another PhD student of mine who came from Thailand. And so what this slide shows is ribotype 017 initially in the top panel has caused lots of outbreaks around the world. So it's traveled a lot, right? It's definitely traveled a lot from Asia. But it's caused outbreaks in not only Asian countries, but it's caused outbreaks in the Netherlands, Poland, Ireland, Sweden, England. So, and then in the bottom panel, you've got a whole bunch of non-Asian countries where there has been some 017, but as I said, it travels a lot, and these are low prevalence places. So what this means, and perhaps I should just mention South Africa. Yes, there's been huge amounts of 017 in South Africa, and it's actually got into the population with TB in South Africa. It certainly got there probably by the ship traffic. All the ships that went from Asia to Europe used to stop in at Cape Town, and that's probably how 017 got there. And my American friends, who I think didn't quite understand what was going on, seemed to forget that in the 1850s, in the gold rush, 300,000 Chinese came to California, and at one point made up 10% of the population in California. And so if you want to know how ribotype 017 got into the United States, and a reasonable number of years ago, my bet would be it came in on one of these boats with all these Chinese immigrants. I also get queries from my American colleagues who say, but, oh, we have all these clade 1 strains in the middle of the United States. Why is that? Well, between 1870 and 1930, 800,000 Norwegians migrated to the United States. That was 1/3 of the population of the country of Norway. They all settled in that Midwest part, Minnesota, although I had 3 very good Minnesotan friends who all worked on C. difficile. And they're all descendant from Scandinavian immigrants. So that's how all those clade 1 strains got into the United States. And of course, as I mentioned earlier, there's the issue of animal movement. And so now with the European Union or European Economic Area or whatever it's called, there aren't any barriers to moving stock around. So this is some work that was published by some animal groups showing that, I think was in, well, it's an estimate from 2019, 1.6 billion livestock transported alive across the EU, and some went overseas. And so that's a problem. If you're going to, like I said earlier, this is the true reservoir of C. difficile. And if you're going to transport animals around, you're going to transport C. difficile. So, and all these animals on here, you can find C. difficile in all of them. And in fact, these days, C. difficile is the most common cause of diarrhea in adult horses. The biggest offenders, I suppose, in contaminating the environment, as we'll see in a minute, unfortunately, is the poor old pig, where high-intensity pig farming uses a lot of antimicrobials. The drug at the moment is a drug called ceftiofur, which is the veterinary equivalent of ceftriaxone, which everybody loved to give to humans, which really did actually amplify C. difficile in humans in the 1980s when ceftriaxone first came out, because everybody in the hospital liked it because you gave it once a day and not 4 times a day. Equine vets love it because they have the equine equivalent, and you've got much less chance of getting kicked in the head by a horse if you're only sticking a needle in it once a day rather than 4 times a day. So this is what's driving this whole problem. So, and going back to the value of doing whole genome sequencing, so this is some more work of Dan Knight's, and if you look at the 3rd column in, or the 4th, so the 3rd and 4th column, one says ribotype, one says sequence type. So this is just a very small group of ribotype 14 strains of C. difficile. Now, the interesting thing about this is, and I mentioned this earlier, that some ribotypes contain more than one sequence type. And here's the classic example, that in ribotype 14, sequence type 49 is only found in pigs. Sequence type 2 is only found in humans. And then there's a sequence type 13, even though the numbers are very small, appears to be found across the 2 groups. And looking at the porcine strains, you'll see they contain many antimicrobial resistance genes. They've got a whole lot of other things. About the only thing the human strains have got is a bacteriophage. They don't have lots of antimicrobial resistance genes. So this is another value of doing whole genome sequencing. So you can look at this sort of stuff. And another example, this is some other work out of Dan's PhD where we sequenced a whole bunch of sequence type 11 C. difficile. And the take-home message here is that for years by ribotyping, we said, if you see on the left there, the list of PCR ribotypes, ribotype 078 and ribotype 126 were supposedly different. If you look at the plot on the right, the big fan looking thing, and you'll see that 078 and 126 by whole genome sequencing are pretty well indistinguishable. So now everybody is lumping them together as one type. So this can be enormously useful. And if you just again look at the outer rings where we have those black patches and purple patches, and yellow versus purple. So this is telling you whether you've got clonal groups. So again, using whole genome sequencing to look at strain relatedness, and in this case, strain origin, whether they're clinical isolates from humans or veterinary or environmental isolates. So I think what happened with the 027 outbreak is that Ceftiofur actually was responsible for amplifying C. difficile Ceftiofur use in animals, was responsible for amplifying C. difficile in the community in the United States. Eventually, those strains, particularly the fluoroquinolone-resistant strains, caused problems in hospitals, and then, as we know, there were those big outbreaks. And we've had similar issues in Australia, with Ceftiofur being licensed for animal use in the 2000s. So let me just get on and I'm going to skip that. What I want to talk about is just this issue in Australia is that the incidence of C. difficile and particularly community-associated C. difficile has been increasing dramatically. We first realised this when we looked at some Australian data from all the states and showed that 25% of the C. difficile infection in Australia was community associated. In the most recent Australian Commission on Safety and Quality in Healthcare report, well, I think it's still the most recent, the significant features highlighted there in yellow. And so the second highlight down, community onset C. diff infection accounted for over 80% of separations. So clearly, as the last highlight says, community onset C. diff infection is now a very significant problem in Australia. And what we see is many of those patients present at hospital emergency departments. But in those hospitals where they do a little bit of investigation of cases, those emergency department presentations are turning out to be community-acquired infections. And so the Commission is trying to publicise this fact that C. diff infection is now a community health issue. So where's it coming from, though? Well, you should have worked out by now that the true reservoirs of C. difficile are animals. Companion animals, food animals, horses, wild animals, reptiles and birds. And I'm just going to give you a couple of examples. This is some European and US data, which just tells you that on the right-hand side there, you can see beef, cattle, but I think this is actually a bigger problem in dairy cows, where Ceftiofur is used a lot. Pork, poultry. Certainly in Australia, poultry don't get antimicrobials, but in the United States they do. So there are plenty of true reservoirs of C. difficile in the United States. And then the problem, as you'll see in a minute, is that manure that comes out of these animals is often used in gardens, home gardens, growing vegetables, market gardens for growing vegetables, and those vegetables are getting contaminated. So I've chosen this one because it's a recent example, and I know you haven't seen this. And we were very excited when we got this published, and that's Natasha up in the right-hand corner there. She actually went out with her husband, I might add, who's a geologist, and so very used to travelling around the middle of Australia. And they collected hundreds of fecal samples from wild horses. You may not know this, but Australia has the biggest population of wild or feral horses in the world, and something like 500,000. And that's a picture that ended up on the front cover of the that volume of applied environmental microbiology that came out in May this year. So what that tells you, if you look at the strains, most of them don't have a ribotype number. Most of them have a QX number. And what that means is that we can't identify them as a strain that people have identified elsewhere in the world. And as it says there, 70% of the strains that she isolated from these wild horses were novel strains. This is just another filler. The picture on the left of horses is another picture she took, which I really liked. I'm also pointing out that we have isolated C. difficile from kangaroos and emus, and that's the Australian coat of arms there in the middle, which has a kangaroo and an emu on it, and they're both animals which we eat, which I should say we don't eat horses though, unlike I think the French. So what about environmental sources? Then once you've got animals pooing out C. difficile, it gets everywhere: soil, water, wastewater, gardens, parks, etc. So a couple of examples. This is another PhD student of mine who's just finished, and so she did this big survey all over Western Australia. Now, Western Australia is a big place. There's a scale down the bottom there you can see. So the distance from up the top of the state to the bottom of the state, I think, is 2,500 kilometres. So it's a long way. So you can go right out into the middle. If you look at that district called the Pilbara district, there's a blue dot there. That is in the middle of nowhere, but you can still dig up dirt and find Clostridium difficile. And she's found the same thing. Lots of C. difficile all over the state. Lots of strains that we don't really know what they are. And of course, there is, I think you've probably seen this slide before, there is this trend these days to use roll-out lawn. And roll-out lawn, certainly in Perth, has grown on pig manure. And so when we've looked at new lawn versus what we call old lawn, 65% of the new lawn has got C. difficile in it. So if you're lying on your new lawn in the summer, then you're probably covering yourself in C. difficile. Eventually, I think the rain washes it all through and it disappears, and you can see that old lawn has significantly less C. difficile. But if you've got brand new lawn, not every state in Australia uses pig manure to grow C. difficile. Some states use chicken manure, which definitely doesn't have C. difficile in it. But it is interesting that the highest rate of community associated C. diff infection in Western Australia is in women of childbearing age, who are the ones that are likely to be home, and they generally live where we have the highest rates. And you can't see this particularly well with this diagram. The highest rates in all the new suburbs, which is where all the new rollout lawn is going. And so this is a recent paper. We have an NHMRC grant at the moment to look at what are the transmission routes of C. difficile. How does it get from those animals, which are clearly reservoirs for it, how does it get to infect humans? And so we've been doing lots of studies looking at where we think you can find C. difficile. And so this is a study of retail vegetables and a whole bunch of households that we randomly chose in both Queensland and Western Australia. And you can see that I think the total was 44% of them had C. difficile in them, and this is chopping boards in the kitchen, in the fridge, unwashed potatoes, which are a big problem. And if you look at the ribotypes down that left-hand column, you'll see the QX ones, which we still can't identify, but there, down the bottom almost, is ribotype 106 that has turned up in Australia, as I mentioned earlier. So this again is an obvious finding that when you go to gardening centres that sell stuff that you put on your garden, composted stuff, it's all full of Clostridium difficile. And we've used whole genome sequencing to show that some of these gardening centre associated strains end up in patients presenting at hospitals. And then eventually it gets into the wastewater, and I know you will have all heard about wastewater surveillance for COVID, because that became very trendy. But you can do the same with C. difficile in wastewater, and this is just a study in Western Australian wastewater treatment plants, where a lot of the water which is used to irrigate sporting grounds in particular is in fact full of C. difficile. And just the kind of one of the prompters for the NHMRC study that I mentioned a minute ago, this again is ribotype 14, C. difficile. And if you look on the right there where it says CG3, clonal group 3, so these are isolates from pigs and isolates from humans with disease. And there you can see a bunch of pig isolates and a bunch of human isolates that are indistinguishable, and they're from different states of Australia. So there's thousands of kilometres. So how does a pig strain, we're assuming it's a pig strain that's got into a human and infected the human. So that was the whole basis of this NHMRC study that we're just in the process of finishing off. How is C. difficile being transmitted? And so what we've chosen to do is use this process that I'll admit I know very little about. It's called system dynamics. And we chose this process to try and plot transmission lines. And so you'll see the next slide I show you looks and is very complicated. So this is a modelling process that uses what's termed systems thinking. It's a very iterative process where we go backwards and forwards with a whole bunch of people. So if you look at the list that I've got there, what's involved in the process, you need to identify various people who kind of had some, as they like to say, skin in the game, who are involved in many of these areas that we think are important. And then you talk to them, and you talk to them about what they do and what they think they know, identify variables and their relationships, and then you build what's called a causal loop diagram. And that's what I'm going to show you next. Then you look at potential feedback loops. The next stage is you turn this into an iceberg model. And as the name suggests, an iceberg model is trying to get at, if you know what an iceberg looks like, there's a little bit on the top and most of it's under the ocean, you can't see it. What you're trying to get at is the bits underneath that you can't see and their relationships. And so you know where possibly you can intervene at a leverage point. So this is our current model. As I said, I told you it was a bit complicated, but you can, let me just break it down into the two main areas. There's the C. difficile in the farm environment, and there's C. difficile in the human environment. And I don't have time to go through all these complicated relationships, but when this paper gets published, not yet, but soon, you'll be able to have a look at it. Suchi and Lim is another one of my postdocs, has kind of turned it into something which is easier to perhaps understand. She likes making these infographics where she puts little diagrams in, but that's just a simplified version of this causal loop diagram. So what does this teach us about infection prevention and control? C. diff infection is not just a hospital issue. In fact, it's probably a minor issue in hospitals these days, but it's a worldwide public health issue. And if you understand a bit about geography, then you can understand what strains of C. difficile will be found in those areas. And knowing the strain, the ribotype, or better, as I mentioned, the sequence type, is actually very helpful in terms of knowing what that strain is likely to do. Certain strains produce more spores, certain strains transmit better, etc. So each jurisdiction likes to know what's going on, or should know what's going on in its own backyard. The problem is there are very few reference laboratories, and indeed in Australia we don't even have a reference laboratory. There's no reference laboratory in Asia. There's a couple of reference laboratories in Europe, and there's certainly a couple in the United States, and certainly one in England. I'm not sure whether, or the UK. I don't know whether the UK is on the cusp of rejoining the EU, but we'll find out soon. There are many sources of C. difficile that are important, and you need to know that. Anywhere there is manure is a problem. Putting antimicrobials into production animals is not a good idea. And the thing we need, certainly in Australia, and I think more around the world, is more studies involving general practice to find out what's going on out in the community. And this, of course, requires a One Health approach. So on that, I'm going to just, if you want to look at a simplified One Health model of what's going on, I would suggest looking at this paper, and I'm sure you'll get a copy of these slides, so you won't need to rush around. This has actually turned into a highly cited publication. Every time you go to a talk on C. difficile anywhere in the world, somebody will pop up this diagram. So it's proved to be quite successful. And of course, I don't do much of this work. All these people here do it. There's my poor emu with diarrhea again. And I certainly like to acknowledge the fact we're doing a lot of work with the University of Queensland at the moment. And on that note, I will say thank you for listening, and I think I'm supposed to hand over to Jane.
Yes, thanks, Tom, very much for that. As usual, an excellent delivery of a fascinating subject. I think you've built really nicely on your previous presentation, where you talked about generalized importance of, community-associated C. diff infection. And clearly now we've moved on from talking about just soil coming in on foodstuffs, but also, of course, the fascinating information about soil and rollout lawns and sports field as well. So that connection is really an essential one, I think, that's being made with the One Health approach. So what would you say if there was an ending sort of funding available would be the priority with this disease globally? Is it the genotyping?
Oh, yeah, I think it is. Well, the two most important things are actually understanding what's going on. I mean, people are still running around typing salmonella. I mean, I know that's important, but as I like to say, and I can only use Australia as an example, how many people, I ask this question all the time of some people in our health department, how many people die of salmonella infection in Australia in a year? Well, it's somewhere between 5 and 7. How many people die of C. diff? Well, it's in the hundreds. So I think and of course, you know, you get one case of someone or this or that or the other, and the health department sends hundreds of people out to investigate. And part of the problem is that C. diff is not notifiable in Australia, and so they don't feel the need to do anything about it. But I think knowing they need to investigate the cases better. But the two most important things: get a reference laboratory set up so that you can do whole genome sequencing, and don't put antibiotics into production animals.
So is there, I think we touched on this last time, is there much work happening in that veterinary space? In relation to
no, there's nothing happening.
Nothing, okay.
That's a problem. I think the equine vets realise that they have created a bit of an issue, because, you know, it's all very well when your $5 piglet dies of, I don't know, maybe I'm underselling piglets, maybe they're worth more than $5. But, you know, when your $20 million racehorse dies, then that's a tricky situation. And I think the industry, the racing industry, which of course certainly in some countries is worth a lot of money, and Australia is one of those countries. And I think you'll find in New Zealand it's a big deal. Certainly in Ireland it's a big deal. In Japan it's a big deal. There are countries where racing is really, really, and the UK of course. So I think that's probably a good place to start, because there's money involved, and you know that if there's money involved, people should take notice.
Yeah, that's a really good context to put it in, isn't it? When you're comparing animals in the food chain, and then of course the financial implications of big cost racing animals. So I really look forward to seeing your publication on the One Health approach. I think that's going to be really insightful for all of us in IPC. And I think those of us that are in countries where we don't see much C. diff don't realise necessarily the impact of it when it will hit us. So more prevention.
I hope you're not including New Zealand as one of those countries.
We don't see a lot of it. Yeah.
Does anybody look?
Well, yeah, I think the testing is happening. The sequencing probably isn't, but yeah, I mean, compared to you took me back in time to when C. diff was evolving in the UK, and I was working in IPC there. That was pretty horrific memories of it there.
Yes.
So compared to that, maybe my it is definitely not as severe. But yeah, I think a really important space to watch, and maybe you've touched on notifiable disease context, and maybe that's the way that we should be.
Well, yes, in England, at least, it is notifiable.
Right.
And in a few other parts of the world, it is. But we haven't gone down that pathway, and I suspect, I don't know whether the new Australian CDC is going to have any impact. My suspicion it won't, because none of the people involved have even heard of C. difficile, I suspect. And whether the US CDC gets back up off its, out of its very parlous state, I have no idea.
Well, there's clearly a lot of work for you still to be doing in this space, I think.
Tell me that. It means I've got to keep working.
All right. Well, thanks again for your contribution, Tom. As always, really enjoy your lectures.
My pleasure. And if anybody listening got any questions, they can email me, of course.
Great, thank you. So there's a slide now with the forthcoming teleclass lectures. Interestingly enough, there's a lecture I see in November on the use of fecal transplant as treatment for Clostridium difficile. So that'll be an interesting follow-on from today's lecture. If we just go to the final slide, just an acknowledgement of our teleclass education patron sponsors. Thanks once again for their support of Weber training. And with that, I'll close for today. So thank you very much for listening in, and thank you, Tom, for your contribution.